release chemicals that affect or attract other phagocytic cells and fibroblasts to the injured area. Here's a picture of the monocytes, again, the largest of the white blood cells. Here we have the lymphocytes, slightly larger than a red blood cell. 20 to 40% of circulating white blood cells continuously migrate in and out of the bloodstream, mostly in organ and lymphatic system and connective tissues other than blood, part of the body's specific defense system. And here we have a picture of the lymphocyte. Here we have three classes of lymphocytes. Again, we indicated, or it was indicated, that these function with the immune system. And types, we have T cell or T lymphocytes, which are cell-mediated immunity, attack foreign cells, or control other lymphocytes. We have B cells or B lymphocytes, which deal with humoral immunity. They differentiate into plasma cells with synthesized antibodies. And then we have natural killer cells, which detect and destroy abnormal cells. A differential count of white blood cell population, you're basically looking at a slide and counting the amount of white blood cells in the area, can detect infection, inflammation, and allergic reactions. White blood cell disorders are leukemia, leukocytosis, leukopenia, with red blood cell production, we talked about erythropoiesis. With white blood cell production, the term is called leukopoiesis, white blood cell production. Hemocytoblasts produce myeloid stem cells and lymphoid stem cells. Myeloid stem cells divide to produce progenitor cells, give rise to all formed elements except lymphocytes. Lymphocytopoiesis, the production of lymphocytes from lymphoid stem cells. White blood cell development. Some lymphoid stem cells remain in the red bone marrow, differentiate into B cells and natural killer cells. Others migrate from red bone marrow to peripheral lymphatic tissues, such as the thymus, spleen, and lymph nodes. To produce lymphocytes, T cells are produced in the thymus. Colony-stimulating factors, hormones that regulate white blood cell populations. We have colony-stimulating factors, which accelerates production of granulocytes, monocytes, platelets, and red blood cells. We have GM, colony-stimulating factors, stimulates granulocyte and monocyte production. We have G, colony-stimulating factors, which stimulates granulocyte production. We have M, colony-stimulating factors, which stimulate monocyte production. And in this picture here, it's basically showing us how we're going from the bone marrow to the hemocytoblastic or hemopoietic stem cells, which separates into a myeloid stem cell or the lymphoid stem cell, and how it goes through its process to ultimately becoming the red blood cell, or in the case, platelets, versus the white blood cells. So that's how it's made. It all starts here, and we can split it out into different areas to become either the red blood cells, the platelets, or a combination of the white blood cells. Here I'd like you to see we've got the red blood cell, platelets, and then we've got 1, 2, 3 white blood cells that are considered granulocytes because they have granules in them. That's the basophil, the eosinophil, and the neutrophil. Agranulocytes are the monocyte and the lymphocyte. The monocyte and the lymphocyte. Platelets. We talked about red blood cells, white blood cells, and now we're going to talk about platelets. Another name for platelet is a thrombocyte. These are cell fragments involved in the clotting system. They circulate for 9 to 12 days. They're removed by phagocytes, mainly in the spleen, 150,000 to 500,000 per microliter of blood. 1/3 of the platelets in the body are stored in vascular organs like the spleen, and they're mobilized during a circulatory crisis. Functions of the platelets: to release important clotting chemicals, temporarily patch damaged vessel walls, reduce the size of break in a vessel wall. The term used for platelet production is thrombocytopoiesis. Thrombocytopoiesis occurs in the red bone marrow. Megakaryocytes, giant cells in red bone marrow, produce platelets by shedding membrane-enclosed packets of cytoplasm. Hormonal control of platelet production is by the hormone thrombopoietin, interleukin-6, and multi-CSF. Hemostasis. Hemostasis, cessation of bleeding, has 3 phases: the vascular phase, platelet phase, and coagulation phase. In the vascular phase, a cut triggers vascular spasm, contraction of smooth muscle fibers of vessel wall, and it lasts about 30 minutes. Vascular phase is indicated. The vascular phase of hemostasis lasts for about 30 minutes after the injury occurs. The endothelial cells contract and release endothelins, which stimulate smooth muscle contractions and endothelial division. Endothelial cells become sticky and adhere to platelets and each other. The platelet phase. Platelet adhesion. Platelets attach to exposed surfaces. Platelet aggregation. Platelets stick to each other, begins within 15 seconds after an injury, forms a platelet plug that closes small breaks. With the platelet phase, activated platelets release clotting compounds, adenosine diphosphate, thromboxane A2, and serotonin, clotting factors, platelet-derived growth factor, calcium ions. As indicated, the platelet phase of hemostasis begins with the attachment of platelets to sticky endothelial surfaces, to the basement membrane, to expose collagen fibers, and to each other. As they become activated, platelets release a variety of chemicals, as we've discussed, that promote aggregation, vascular spasm, clotting, and vessel repair. Factors that limit growth of platelet plug. Prostacyclin inhibits platelet aggregation, inhibitory compounds released by white blood cells. Circulating enzyme breaks down ADP, ADP. Negative feedback from serotonin, etc. Blood clot isolates the area from general circulation. Those are the factors that limit growth of the platelet plug. The coagulation phase, or blood clotting, begins 30 seconds or more after an injury. Depends on clotting factors, procoagulants, calcium, and 11 different proteins. Proenzymes, inactive enzymes convert to active enzymes that direct reactions in clotting response. The coagulation phase involves chain reactions of 3 pathways: the extrinsic pathway, intrinsic pathway, and the common pathway. The extrinsic pathway, damaged endothelial cells or peripheral tissues release factor 3, tissue factor. Enzyme complex activates factor X. The intrinsic pathway begins with activation of proenzymes exposed to collagen fibers at the injury site. Platelets release PF3. Enzyme complex activates factor X. Common pathway It begins with activation of factor X. Factor X activates prothrombin activator, converts prothrombin or proenzyme to thrombin. Thrombin converts fibrinogen to insoluble fibrin, producing a blood clot. This picture is showing you the three pathways, the extrinsic, the common, and the intrinsic. Extrinsic coming this direction here, factor 3, changing to the factor X. Here we're coming through the intrinsic to the factor X as we come to the common pathway. Coagulation or blood clotting involves a complex sequence of steps leading to the conversion of circulating fibrinogen, a soluble protein, into fibrin, an insoluble protein. As the fibrin network grows, blood cells and additional platelets are trapped in the fibrous tangle, forming a blood clot that seals off the damaged portion of the vessel, as everything is coming down from fibrinogen to fibrin. Fibrinogen to fibrin, closing off that damaged area. Feedback control of blood clotting. Anticoagulants, enzymes that inhibit clotting, such as antithrombin 3. Heparin accelerates activation of antithrombin 3. Thrombomodulin activates protein C, which inactivates clotting factors and stimulates formation of plasmin. Prostacyclin inhibits platelet aggregation and opposes factors. Calcium ions and vitamin K are essential to the clotting process. All three pathways require the calcium. Vitamin K is required for synthesis of 4 clotting factors. Some terms given to bleeding and clotting extremes: thrombocytopenia, hemophilia, thrombophilia, deep vein thrombosis. Clot retraction makes reference to it pulls torn edges of the vessel closer together, reduces residual bleeding, stabilizes the injury site, reduces size of the damaged area, making it easier for the fibroblasts, smooth muscle cells, and endothelial cells to complete repairs. Clot retraction: once the fibrin meshwork has formed, platelets and red blood cells stick to the fibrin strands. The platelets then contract, and the entire clot begins to undergo clot retraction, a process that continues over 30 to 60 minutes. Term fibrinolysis: gradual process of dissolving a clot. Thrombin and tissue plasminogen activator. Activate plasminogen, producing plasmin. Plasmin digests fibrin strands. And that concludes Chapter 19, The Blood.